Hi, I'm Susmita, working in Dr. Kufal lab as summer research intern and Khorana Scholar 2012 in University of Iowa. And my topic for this summer is role of TXNIP as a vital gene in leukemia treatment sensitivity. Glucocorticoids are a part of the standard treatment for BCP-ALL, which is B-cell precursor acute lymphoblastic leukemia, and synthetic glucocorticoids like dexamethasone and prednisone are used for the treatment. This cancer is most prevalent. This is the most prevalent cancer among children below 10 years of age, and though the standard treatment is remarkably effective, 10% of the patients are resistant to the treatment. Importantly, which children will respond to the treatment and which do not respond to the treatment can be predicted whether isolated leukemia cells can be killed by glucocorticoids. In this cancer, leukemic blasts are formed instead of mature white blood cells. So what we are interested in looking at is how to resensitize the resistant patients. Glucocorticoid receptor is a ligand-activated transcription factor. The glucocorticoids, when they enter the cell, they bind to the glucocorticoid receptor, and the GCGR complex then translocates to the nucleus and binds to the glucocorticoid response element and causes either transactivation or transrepression. We are looking at genes which are critical to the glucocorticoid ability to induce apoptosis, which is programmed cell death. Which are regulated in the sensitive patients but unregulated in the resistant patient. We term these genes primary positive glucocorticoid regulated genes. To understand how GR regulates these genes and why it fails in some of the resistant patients, we seek to identify GRE, which is the glucocorticoid response element associated with these genes. The gene I have chosen to work on is TXNIP. Is TXNIP primary causative glucocorticoid regulated gene? Gene expression microarrays on 4 sensitive cell lines, 1 sensitive patient sample and 1 resistant patient sample, measuring the response of each to dexamethasone, yielded 50 genes which were common among the sensitive patients that were unregulated in the resistant patients, and TXNIP is one amongst these genes. In addition to being regulated at an early time point, we found that GR is bound near to an upstream of TXNIP gene, TXNIP in BCP-ALL cell line NAM6. There's also some evidence that TXNIP is associated with apoptosis in other systems by inhibiting the glucose uptake, by allowing the accumulation of reactive oxygen species, and stimulating the activity of apoptosis signaling kinase and arresting the cell cycle. These are some evidence which show that TXNIP could be a positive gene. Chromatin immunoprecipitation followed by deep sequencing of these NAM6 cells reveals 4 potential glucocorticoid receptor binding regions GBR1, 2, 3 and 4, which are upstream of the TXNIP gene. So which of these binding regions actually plays a role in the gene expression is what I will be doing, what I did this summer. Our model is that in BCP-ALL, the glucocorticoid receptor binds to the GBR, which is the glucocorticoid receptor binding region, which is near the TXNIP transcription start site inducing gene expression, and expression of TXNIP induces apoptosis in BCP-ALL cells. So how to test the activity of the GBR? Testing each region by mutating the genome is both expensive and time consuming. So we decided to do a luciferase assay by isolating each GBR from the genomic region, doing a PCR of it, and engineering restriction sites into each end, and cloning each GBR into a vector using these restriction sites. After testing for the presence of the insert in the plasmid and sending it to sequencing, we transfect the plasmid into B1, which is the sensitive cell line, and RAH, which is the resistant cell line. GBR activity is then assayed by measuring how much luciferase activity changes in response to the glucocorticoids. To ensure that the change is due to the GR and not because of the vector, they've used 2 vectors like PGL3 and PGL4.26. Here you can see that this is the multiple cloning site in which the insert will be there. This is the promoter region, this is the luciferase gene. So coming to the experiments and results. First, I've done cloning using single restriction site, and the primers were designed using Primer 3 software, in which I couldn't isolate GBR3. So what I've done in this next step was doing the primer design for GBR3 by hand and double digestion for GBR3, but the rest remained the same as in the first set of experiments. the first set of experiments and the second set of experiments which I did with single restriction site did not yield positive clones. So what I have done was, but the double digestion of using GBAR3 yielded positive results. Using the success of this method, I have redesigned my primers by hand to encompass the whole of the genomic region and using double restriction sites for even GBR1, 2 and 4. So when I have done that, I have got 7 out of 8 constructs of the vector and the insert which were rightly sequenced. So this is the restriction digest screening gel, in which this is the PGL3 and these are the red ones show the positive ones. So I have 7 out of the 8 constructs. The future experiments are transfection of the plasmid into B1 cell lines, which is the sensitive one, and REH, which is the resistant cell line. These experiments will establish whether TXNIP is a primary gene. But what we also need to check is whether TXNIP is a causative gene. To do this, we have been provided constructs from Dr. Don Ayer lab at University of Utah, which both overexpress and knock down TXNIP. Overexpression of TXNIP should cause apoptosis in the cell, and knockdown of TXNIP should give resistance to glucocorticoids. So by doing these experiments, we can establish whether TXNIP is a causative gene. So that's about my research work, and I'm very thankful to Dr. Miles Bufal, who has been very helpful for me this summer, and Dr. Don Shenton, who has helped me in these experiments, and Dr. Ernie, who was the coordinator of the summer program here in the University of Iowa. Thanks to Khorana program, USSTF and Department of Biotechnology for the funding and for sponsoring us. Thank you.